This is my video blog 3 regarding neoevolution. Feinberg describes the concept of neoevolution in his 2011 TED Talk, indicating that this new type of evolution is not simply natural. To begin his talk, Feinberg inquires with his audience regarding different aspects of the genetic capacity, such as creativity, memory, and longevity. He asks them if they could choose, which of these aspects would they choose to improve? Darwin's theory of evolution indicates that the most ideally adapted population survive via the process of natural selection. In contrast, neoevolution employs that it is the individuals that guide and choose evolution's progression. An example of this self-directed evolution is the choice of some cultures to have more male children than female. According to Feinberg, neoevolution proposes the possibility to accelerate evolution and compress its timeline significantly. Technology is unquestionably a driving force of this neoevolution. In Paul Wolpe's TED Talk titled "It's Time to Question Bioengineering," he speaks about the title science. Bioengineering is allowing scientists to alter the physiological forms on our planet. This technology has resulted in the many different dog breeds that we're familiar with today, as well as creation of new hybrid animals such as the liger. Wolpe indicates that by taking control of our evolution and manipulating nature in this way, we must also consider the ethical implications. We need to take responsibility for the choices that we make, as they will affect the type of bodies as well as the type of lives that we have in the future. A subcategory of bioengineering is genetic engineering. According to yourgenome.org, genetic engineering, also known as genetic modification, is the process of altering the DNA in an organism's genome. Currently, genetic engineering is being used to study Alzheimer's disease, among other things. The APP gene has been identified as a characteristic of people with this disease. To study its affiliation with Alzheimer's, researchers genetically engineered the nerve cells of the nematode worm to contain the APP gene, effectively giving the worms Alzheimer's. The researchers also tagged the APP protein with a green fluorescent protein so that the cell could be easily identified. As a result, the progression of Alzheimer's disease and the cells it's affected with in the worms can be tracked, and this information can be applied to understanding the progression of Alzheimer's in human patients. In addition to its current applications, the potential benefits of the technology include altering the genomes of human embryos to eliminate genes that cause disease. This could potentially decrease human suffering, but also brings up the question of ethical limitations. Will we as a society set limitations to the use of this technology, or will we take it too far? This also raises the question about the future generations affected by the technology in the reproductive process. Is it fair for the current generation to manipulate the future? I think that there is a necessity for regulations if we are to continue the use of this technology. And while I can understand the concern of its potential abuse, the technology's potential to improve current and future lives is invaluable and cannot be ignored. Therefore, I do think that we should continue to use the technology, but also acknowledge that we need to have limitations and have the proper regulations in place. This concludes my video blog 3.